Phycology, the study of algae, parasitology, the study of parasites, and virology, the study of viruses and how they function inside cells. These broad areas encompass a number of specific fields. These fields include immunology, the study of the immune system and how it works to protect us from harmful organisms and harmful substances produced by them. Pathogenic microbiology, the study of disease-causing microorganisms and the disease process, epidemiology and etiology, microbial genetics, which is linked to the molecular biology. Food microbiology, studying the effect of food, spoilage, and so on. The microbiological discipline of relevance here is pharmaceutical microbiology, an applied branch, and microbiology, once considered as an offshoot of industrial microbiology but now a distinct field. Pharmaceutical microbiology is concerned with the study of microorganisms associated with the manufacture and pharmaceuticals. This is with either using microorganisms to help to produce pharmaceuticals or with controlling the number in a process environment. This latter concern is about ensuring that the finished product is either sterile or free from those specific strains that are regarded as objectionable. The extends through the manufacturing process encompassing starting materials and water. Pharmaceutical microbiologists are additionally interested in toxins, microbial byproducts like endotoxins and pyrogens, particularly with ensuring that these and other vestiges of microorganisms which might elicit adverse patient responses are absent from products. Biological contamination becomes a problem when it results in unwanted effects occurring in pharmaceutical preparations. In drawing from risk assessment terminology, pharmaceutical microbiology centers on understanding the likelihood of product contamination arising, understanding the severity of such contamination, considering ways to minimize contamination, and where contamination cannot be satisfactorily mitigated, using established and developing now methods to detect contamination. To understand the severity, it is necessary to understand the type of product, it is intended use, and the nature of numbers of contaminants. Microbial contamination of sterile injectable products or parenterals presents the greatest risk, for this may lead to death of patient. Whereas with other products, aromas, off flavors, or discolorations caused by microorganisms may have fewer adverse effects. Therefore, with respect to the sterile products, the main concern is with any potential microbial contamination with non-sterile products such as inhalations, tablets, oral liquids, creams, and ointments. A level of microbial contamination may be tolerated with a greater concern centered on the presence or absence of objectionable microorganisms. The types of microorganisms of concern depend upon the type of product and route of administration. The foundation of pharmaceutical microbiology is use of culture media. These are nutrients and growth factors designed to cultivate and grow bacteria and fungi. This foundational basis is being partially eroded through the increased use of rapid and alternative microbiological methods. Although even here a substantial number of these methods remain growth-based, the overriding technique that the microbiologist must apply is an aseptic technique, the prevention of contamination during the manipulation of samples and cultures. The growth of microorganisms and the maintenance of aseptic technique require trained and skilled microbiologists. This chapter, as a way of short introduction to the various texts in this book, outlines the essentials of pharmaceutical microbiology, many of which are discussed in detail throughout this book. Microbiology may be divided into microbiology of the culture yeast used in the process and other microbes, broadly termed contaminants. The latter group contains bacteria, wild yeast, and molds, but not pathogenic microorganisms, as they do not survive or grow in the final product. Microbiology in brewing is primarily a problem of economic loss through spoilage. This would lead to a loss of consumers if an off-flavored product were marketed. Microbiological methods are also published by the organizations mentioned previously. Prevention and control microbial problems are carried out by extensive cleaning and sanitation programs in combination with intimate knowledge of the engineering, design, and process operations in brewing process. Although in-place cleaning systems have revolutionized plant hygiene, careful attention to detail. Although in-place cleaning systems have revolutionized plant hygiene, careful attention to details, and cleaning and sanitation are essentials for the prevention and control of microbiological contamination. Cardiovascular pathology. Microbial diversity can be defined as the range of different kinds of organisms. Different microbes thrive throughout the biosphere, defining the limits of life and creating conditions conducive for the survival and evolution of other living beings. The different kinds of microbes are distinguished by their differing characteristics of cellular metabolism, physiology, and morphology, by their various ecological distributions, and activities by their distinct genomic structure, expression, and evolution. The diversity of microbes presently living on Earth is known to be high and is thought to be enormous, but the true extent of microbial diversity is largely unknown. New molecular tools are now permitting the diversity of microbes. New molecular tools are now permitting the diversity of microbes to be explored rapidly and their evolutionary relationships and history to be defined. The purpose of this article is to define the scope, highlight major themes in our current understanding of microbial life, and to describe recent progress in expanding knowledge of the evolution and biological significance of these organisms. Types of production. Microbiota present in wheat and semery processing. The microbial diversity of coffee beans in wheat processing is smaller compared with that in dry processing because the fermentation time is shorter, up to 48 hours. Up to 48 height, and there is a more rapid pH decline from 6 to 4.3. This microbiota comprises few bacterial and yeast species, there is no report of filamentous fungi involved in wheat fermentation. The mucilage adhered to beans. Beans is the substrate used by bacteria and yeast during fermentation. Higher microbial diversity in washed coffee fruits could be observed using molecular identification techniques that allow the detection of cultivable and uncultivable microorganisms. The molecular identification of microorganisms associated with coffee showed that in cemetery process, the microbial succession occurred. Bacteria dominated at the beginning of the process, but after 72 hours, the bacteria and yeast populations reached similar values, average 10 to the power of 6 CFU G-1, and after 192 hours of fermentation, the yeast population predominates. Currently, there is still doubt regarding the apparent bacterial